Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. My dear students, today inshallah we are going to answer the questions of Al-Azhar Model Exam 2020. The first question, A, write the scientific term. Number 1, chemical substances that are present in healthy and infected plants which recognize the presence of a pathogen and activate the plant defense against it. Of course, they are the receptors. Right. Number 2, non-coding genes present at the ends of the chromosomes. They are the satellite DNA parts. Two pieces of DNA connected together by ligase enzyme. Two pieces of DNA of different living organisms. Of course, it's the recombinant DNA. The non-specific defense mechanisms in the area of injury as a response to damaging of the tissues caused by the injury or the infection. Of course, this is the inflammation, which is considered from the second line of defense. Number 5, corresponding base pairs in the DNA molecule, the complementary nitrogen spaces. Number 6, all the DNA of one cell of an organism, or the total number of genes in one cell of the organism, is the genome. Number B, give reasons for each of the following. Number 1, cell division indicates that chromosomes are the bearers of the genetic information or responsible for the transfer of genetic information from parents to offspring. That's why. Because the chromosomes are composed of proteins and DNA, which consist of genes that represent the unit of the genetic information. During the cell division, the chromosomes split from each other to produce new cells which receive a complete copy of the original cell's genetic information. Number 2, thymosin hormone plays a role in the immune system. That's because it stimulates the maturation of the T cells and their differentiation into the 3 types of T cells, which are the T helper, the T suppressors, and T cytotoxic cells. 3, there is a common plan for protein synthesis in all the living organisms because there are 20 types of amino acids participate in the building of the proteins. All of them have the same basic structure, as you know, but they only differ in the alkyl group, and they bind together forming the different types of proteins according to, yes, the type, the order, and the number of amino acids. Number 4, the number of T suppressors or TS cells increase after the elimination of the pathogen to secrete the proteins which are called the lymphokines, which suppress or inhibit the immune response or stop it. Therefore, the plasma cells will stop producing the antibodies, and many of the T helper cells and the activated T cytotoxic cells will die, but of course some of them will be stored in the lymphatic organs where they stay ready to combat any similar infection in the future when needed. Number 5, tRNA or transfer RNA molecules can be transferred safely between the cells of different organisms. Why? Because all the types of tRNA have the same codes in all the cells of different organisms. As you know, the genetic codes are universal. C. Compare between each of the following. Number 1, the chemokines and the interleukins. The chemokines are the chemical substances which recruit or guide migrate the large circulating phagocytes, which are found in the blood with large numbers, to the site of the existence of the microbes or the foreign particles to prevent their spreading or reproduction, while the interleukins, as you know, their function is to mediate, communicate between different immune cells and between the immune system and different body cells. Of course, they help the immune system to perform um, the defense mechanisms secreted by the T helper cells to activate. During the humoral immunity, they activate the B cells to divide and differentiate into the plasma B cells, which produce the large amounts of antibodies, and the memory B cells, which keep the information of the antigen for the coming infections. And during the cellular immunity, they activate. Number two, the nucleotides and the ribonucleotides. As you know, the nucleotides are the building units of the DNA or deoxyribose nucleic acid. It contains pentose sugar called deoxyribose sugar. The nitrogenous bases are adenine, guanine, thymine, and cytosine. Of course, they form the DNA double strands, while the ribonucleotides, they are found in the RNA single strands. They contain ribose sugar, and there is another difference in the nitrogenous bases. They are adenine, guanine, cytosine, and uracil instead of thymine. Question 2a. If nitrogenous bases sequence of nucleotides on one strand of a gene is, as you see, then write the sequence and write the complementary strand. Of course, we are going to write the sequence again from 5' to 3'. Then we will write the complementary on the other strand. As you know, C or cytosine always base pair with guanine, and thymine always base pair with adenine, as you know. And the two strands are anti-parallel from 5' to 3'. The other, or the complementary, starts from 3' to 5'. Number 2, does this sequence represent a code for the protein synthesis or not? Explain. Of course, we are going to examine the sequence. What is the answer? Of course, no, it doesn't represent the code for protein synthesis. Why is this? Because the 3' to 5' strand doesn't carry the code transcribed to the start codon, which the start codon is AUG, and also it doesn't contain any of the 3 stop codons, UAA, UAG, and UGA. If this gene doesn't represent a code for protein synthesis, what are the modifications that can be performed to its sequence to represent the protein synthesis code? And what is the number of amino acids resulting from this gene after modification? Of course, the modification is we are going to add the start codon to the strand at 5' direction and the stop codon at 3' direction. Then write the complementary from 3 to 5. Then we transcribe from the complementary one from 3 to 5 the messenger RNA. This is the messenger RNA transcribed from 3' to 5' as strand. Then we should add the AUG and one of the stop codons. Of course, the number of the amino acids are 6. Okay. patch, then rewrite the complete sentence. The answer is, of course, number 1, the skin, D, covered with tough horny layer. Cerumen, number 2, fits for number E, secreted from the ears and kill the microbes. Number 3, tears contain antimicrobial substances which kill the microbes. Number 4, the mucus of the respiratory tract fits for F, trap the microbes and foreign particles entering with the air. Number 5, saliva contains substances that kill the microbes and enzymes that dissolve them. Number 6, the acidic gastric secretion contains hydrochloric acid which kills the bacteria. C1. What is the function of each of the following? Pairs patches, nucleolus, polyadenine tail, and repetitive gene. Pairs patches, as you know, doesn't have a known function up till now, but they play a role in the immune response against the pathogenic microorganisms which enter the intestine. The nucleolus is very important for the production of the ribosomes by 4 types of ribosomal RNA with 70 polypeptide chains. The polyadenine tail, which is present in the terminal part of the messenger RNA, protects the messenger RNA from the action of the enzymes in the cytoplasm. And the repetitive gene, of course, we know that it's very important as they are found in hundreds of copies needed to synthesize the ribosomal RNA and histone proteins. They are needed in large amounts. The multiple copies of the genes, which is responsible for the formation of each of the following. Number 1, the tyloses. The tyloses are produced by the living parenchymatous cells adjacent to the xylem vessels and tracheids. Of course, they are overgrowths which protrude through the pits inside the xylem vessels and tracheids to prevent the spreading of the pathogen that enters the xylem vessels. Number B, the lymphatic toxins. Can you remember? Yes, they are secreted by the cytotoxic T cells during the cellular immunity. Number C, peptide bond during the protein synthesis. The peptide bonds are produced through the peptidyl transferase reaction by the help of the peptidyl transferase enzyme, which catalyzes the reaction in the peptidyl part or the P site of the large ribosomal subunit. Reverse transcriptase, as you remember, it's the enzyme that produced by the viruses with RNA genome, which use it to convert their RNA genome to DNA that can be joined to the host's DNA genome. Question 3. A. Correct the underlined words. Number 1, cephalosporin, canvenine, and glycosides are the chemical substances that are used to induce mutation. The correct answer is mustard gas, colchicine, and nitrous acids. Number 2, the histamines are secreted by the B cells and the natural killers. This is incorrect. They are secreted by the mast cells and the basophils. Cork is secreted in plants to isolate the injured areas. The correct answer is the gums. Lysis is one of the most important antibody mechanisms to resist the viruses. You know that to resist the viruses, there is another mechanism, which is the neutralization. The least number of tRNA molecules needed to build the polypeptide chain formed from 150 amino acids of 19 different types is 148. You know that we can count the number or the least number of tRNA molecules according to the number of the types of amino acids, not their number. So the correct answer is 19. The sequence TACCT is repeated 100,000 times in the middle of one chromosome in Drosophila insect. The correct sequence is AGAAG. Number B, 1. What is meant by each of the following? Genetic code. It's a particular sequence of nucleotides on the DNA which are transcribed to a complementary sequence on messenger RNA to be translated into a particular sequence of amino acids in a polypeptide chain that makes a particular protein. What is meant by complements? They are protein substances which destroy the microbes in the blood after their conjugation with the antibodies. They lyse the membranes of the antigens and dissolve their contents, which makes them easily engulfed by the phagocytes. Number 2, fill the following spaces. As you see, there are spaces, but you can fill them easily by examining the complementary ones. As you see here, we can find the answer easily, as all the time we will have the complementary one to find. Explain the form in which the DNA molecules are present in the chromosomes as revealed by the electron micrograph and biochemical analysis. The first step to pack the DNA inside the nucleus in the form of chromosomes. DNA strings are wound around clusters of the histone molecules, histone protein molecules, forming strings of nucleosomes. This shortens the molecules about tenfold, but it must be packed about 100,000 times more tightly to fit into the nucleus. Then the strings of nucleosomes are coiled to pack nucleosomes together. The tightly coiled strings of nucleosomes are arranged in large loops which are held in place by the structural non-histone proteins of the chromatin, and the chromatin packed up as tightly as possible by more non-histone proteins to be condensed. And you remember that the condensed chromatin are composed of DNA, histone, and non-histone proteins. Then before the cell division, they are distinguished into the form of chromosomes. Now let's start with question 4a. What happens in each of the following cases? Number 1, a young man is affected by bone marrow cancer. You know that the bone marrow is responsible for the production of the all the blood cells, and in particular here the white blood cells. So this will affect the production of white blood cells as the lymphocytes, so there will be no production of the antibodies which defend the body against the diseases. Number 2, the arrangement of the genes on a chromosome is changed. This will lead to a chromosomal mutation due to the change in the structure of the chromosome. Number 3, the absence of antigens from the surface of the microbe. Of course, the immune system cells will not recognize this antigen, so they cannot find the suitable defense mechanism to defend the body against this microbe. Number 4, Tag polymerase enzyme in PCR apparatus is replaced by the DNA polymerase. So the production of the DNA pieces by using the gene machine will stop due to the absence of the Taq polymerase enzyme, which works at a high temperature to produce 1,000 pieces of DNA per minute. Number B, 1. If a blood sample contains 10,000 white blood cells, calculate the following number in the sample. Of course, we have revised all the rules needed to answer this type of questions in the previous online lecture, as you remember. Number A, the average of lymphocytes 10,000 times 25% equal 2,500 lymphocytes. Number B, the maximum number of B lymphocytes. So we need to calculate the maximum number of lymphocytes first, then the maximum number of B lymphocytes, which is represented by 15%. So the maximum number of lymphocytes equal 10,000 times 30% equal 3,000 lymphocytes. The maximum number of B lymphocytes 3,000 times 15% equals 450 B cells. The least number of natural killers, so we are going to calculate the least number of lymphocytes. 20% equal 2,000 lymphocytes. Then the least number of the natural killers, 2,000 times 5% equal 100 cells. The number of T lymphocytes, and they didn't mention if they are asking about the maximum, minimum, or the average, so we are going to calculate all of them. The maximum number, 3,000 lymphocytes times 80%. The minimum, 2,000 times 80%, and the average of T lymphocytes is 2,500, the average number of lymphocytes, times 80%. Number 2, draw a labeled diagram for the messenger RNA molecule. As you see, it's very easy. In the beginning, here is the ribosomal binding site. Then comes the codons. It starts with the AUG, the start codon, and the last one is the stop codon, which is one of three: UAG, UGA, and UAG. And there is the polyadenine tail, which protects. Number C, if you have the following sequence of DNA, as you see, write down the complementary sequence. This is the complementary sequence from 5' to 3'. If you have a restriction endonuclease enzyme with a recognition site, as you see here, this is the recognition site of the restriction enzyme. Number 1, draw the arrows to illustrate the position of action of the restriction enzyme. These are the two strands, and these arrows show the site of action of the restriction enzyme. Number 2, what is the result of action of the restriction enzyme on the recognition site? Of course, as you know, the restriction enzyme recognizes the recognition site and cuts near or at it. This produces a staggered cut, in which the double helix is left with two single-stranded ends, which are called the sticky ends, which can base pair later on with any single strand produced by the same enzyme. Question 5. A. Study the following figure, then answer. As you see, the figure belongs to the steps of the protein synthesis. The first question is, what is the stage of protein synthesis illustrated in the figure? It's the termination stage where the protein synthesis stops. Number 2, write the labels. 1 is the produced protein or the polypeptide chain. Number 2, the messenger RNA. Number 3, the small subunit of the ribosome. And number 4 is the stop codon. Question 3, explain the reason of the presence of the scissors in the figure. As you see in the figure, the scissors are found here to cut the start codon, which codes for methionine. So the scissors in the figure cut the AUG start codon. That means the produced protein doesn't start with the methionine amino acid. As choose the right answer. The direct evidence of DNA structure obtained for the experiments performed by Hershey and Chase, Franklin, Corona, or Watson and Crick. The answer is Franklin. Number 2, from biochemical plant immunity, deposition of gums, cork formation, phenol secretion, or tylosis formation. The answer is phenol secretion. Each of the following sequences can be the anticodon of valine amino acid except one, which is the AUC. Number 4, if adenine ratio in one strand of a double helix is 10%, then the percentage of thymine in the same strand could be, yes, may not equal 10%. As you know, the ratio of adenine equals the ratio of thymine in the double helix, in the 2 strands of the DNA double helix, not in one strand. Number 5, all the following organs are lymphoid organs except we have skin, tonsils, thymus gland, and Peyer's patches. Of course, the answer is the skin. Number 6, the plasmids are not found in bacteria, amoeba, chloroplasts, or yeast. The answer is amoeba. If 3 molecules of DNA are placed in 3 different test tubes, deoxyribonuclease enzyme was added to the first one, DNA helicase was added to the second tube, and RNA polymerase was added to the third tube. Mention the action of each enzyme provided that suitable conditions are available. Test tube number 1, where the deoxyribonuclease enzyme is added. Of course, the effect of deoxyribonuclease is the DNA is completely hydrolyzed. Test tube number 2, which contains the DNA helicase, it will break down the hydrogen bond between the 2 strands and separate the 2 strands of DNA. In test tube number 3, where RNA polymerase is added, it will transcribe a complementary strand of messenger RNA from the strand that contains the promoter, which is the strand from 3 to 5. Number 2, write the number that indicates each of the following. Number of genes in a human cell, 60,000 to 80,000 up to now. Number 2, the percentage of genes of unknown functions in an eukaryotic cells. That means the non-coding genes, they represent more than 30%. The number of disulfide bonds between the heavy chains in a single IgM antibody. 10. The number of polymerase enzymes in the eukaryotic cells, we have 4 types, DNA polymerase and 3 types of RNA polymerase. The number of ribosomes that can bind to a single messenger RNA molecule during the process of translation, they may reach 100 ribosomes. Number 6, the number of adenine and guanine bases that may be lost from DNA in one day. The number is 5,000. That's all for today. Please study well till we meet again in the next online lecture to answer Al-Azhar 2019 exam. So be ready. Thank you. Wassalamu alaikum wa rahmatullahi wa barakatuh.